Hello, my name is Dr. Osvaldo Padron, and I'd like to thank the International Continence Society for allowing us to present our results of the Sans-Uui feasibility study. These are my disclosures. The Sans-Uui trial is a single investigational device exemption study that includes both a feasibility and a pivotal phase. Today I'm reporting on the preliminary results of the feasibility phase of this trial. A total of 39 subjects were enrolled across 11 centers, 8 in the United States and 3 in Europe. The goals of the feasibility study were to confirm system performance, evaluate subject compliance, and to determine daily stimulation duration. The Neuspera implantable sacral neuromodulation system consists of a miniaturized implantable neurostimulator that is 52 millimeters long and 2.3 millimeters in diameter. The neurostimulator combines a receiver antenna, a hermetically sealed implantable pulse generator, and a timed stimulation array with 4 programmable electrodes. The neurostimulator antenna receives energy from an externally worn powering unit, the wireless transmitter that is held in place over the sacrum in custom undergarments. The neurostimulator is placed in S3 or S4 foramen in a single procedure. A small incision was made and a 7-French introducer dilator is placed. The neurostimulator comes preloaded on the end of a push rod. The push rod and the neurostimulator are placed through the introducer, and the push rod is then retracted to deploy the neurostimulator. The preformed curve of the electrode array follows the nerve bundle anatomy. Once the neurostimulator is placed into position and confirmed, the introducer is removed. The incision can be closed with a stitch or dermabond. The learning curve for this implant is very similar to neurostimulators that are commercially available. The feasibility phase was designed to determine the number of hours of daily stimulation duration required to maximize effectiveness in the pivotal phase of the trial. The design included a trialing period with daily stimulation titration phase of 2, 4, and 8 hours to determine the daily stimulation duration required to achieve a 50% or greater reduction from the baseline of UUI episodes. Screening 89 subjects yielded 39 subjects enrolled and 34 subjects implanted. At the end of the trialing phase, 97% of the responders were using 2 hours of stimulation, with just 1 using 4 hours. The inclusion and exclusion criteria were similar to those used with SNS pivotal trials of commercially available therapies. The baseline demographics show a mean of 62 years old, a BMI of 30, and 95% of the enrolled subjects were female. The graph on the left, you'll note that 90% of the subjects at 6 months had a 50% or greater reduction in their baseline UUI events per day, and 36% of the subjects were 100% dry at the 6-month visit. In the graph on the right, the average reduction of UUI events across all subjects was 81% at the 6-month visit compared to baseline. The graph on the right also shows the reduction of UUI events from baseline at 12 months. The statistical significance was demonstrated at all points from the end of trial visit out to the 9-month visit. Subjects are still continuing through the follow-up phase, and only 7 of the patients have completed the 12-month follow-up visit to date. The quality of life composite score and all the component scores from the IC/IQ OAB quality of life tool demonstrated improvements to 3 to 4 times greater than the 10-point change required to demonstrate a clinical significance. Quality of life measures were aligned with the responder rates at the 6-month follow-up visit. The data from the Sans-Uui study showed 0 infections, 2 neurostimulator dislodgements in 2 subjects, 3 device fractures in 2 subjects. The device fractures occurred at the base of the electrode array. One subject required an explant, and there were no serious adverse device events. Implant site pain was reported in 5 subjects. 4 of the documented as the postoperative healing without sequela. One of the patients was my patient that had a fractured device. It is worth noting that device removal is very similar to other SNS commercially available. There were 4 successful revisions. 1 subject is awaiting a revision. The majority of adverse events occurred within the first 50 days from implant, including 2 of the 3 device fractures. The manufacturer has made changes in the instruction for use with respect to the implant technique, as well as created a shorter implant for use in the pivotal trial. And the manufacturer is also reinforcing the base of the electrode array as a secondary measure to mitigate fractures of this device. In conclusion, therapy effectiveness of the feasibility phase demonstrated that 90% of subjects experienced greater than a 50% reduction in UUI episodes at the 6-month visit, with only 2 hours of stimulation. Quality of life composite scores of all components demonstrated a 3 to 4 times greater than the 10-point change needed for clinical significance. 36% of the patients were 100% dry at the 6-month visit. These results are consistent with CAN and lead stimulations that utilize 24/7 stimulation. The Sans-Uui study had 0 infections, demonstrated safety consistent with commercially available products, and the manufacturer has made changes to address device fractures and dislodgements observed during the feasibility study prior to initiating the pivotal phase. Thank you.